Thank you, everybody, for being here today. I apologize because I'm having some sore throat. Okay, Oryzon is basically developing epigenetic drugs in the oncology and CNS indication, and we are publicly listed in Spain. So I need to draw your attention to this legal notice. So as I said, we are basically an epigenetic company, and we are basically building our all programs around one of the most fascinating targets, which is LSD1, which is an histone demethylase, a chromatin modifier, but also a master transcriptional gene. So we have, I would say, an unicity because there are some other companies, some other interesting epigenetic companies in the oncology field, and we are also very active there, and I will show you our program in a minute. But I think that we are the one and only that is developing also this in CNS, where epigenetics is certainly going to play an interesting role in the next years, in the years to come. So these programs are in these two main indications. In terms of company, we are based in Madrid, but we have a very strong imprint in the US with our medical team in Boston, activities in San Diego, and BD in New York. The company has a solid financial situation right now. We were recently setting up a convertible notes program. We have an expected runway till mid of 2024. So as I said, we have an epigenetic program with two pillars around LSD1 in two different indications, CNS, oncology, with two uncorrelated assets, both of them in phase 2, both of them have been de-risked, both of them have been shown in human patients to provide clinical benefits. I am highlighting this because we think that these are probably the most immediate investment thesis in the short midterm, but the program, as you will see, is deploying in a very ambitious manner. So starting by oncology, so we got recently granted by the FDA the orphan-drug designation for small lung cancer. We had got that before for AML in Europe and in the US. And we have got recently a very interesting agreement with the National Cancer Institute in the US, a CRADA agreement to explore different indications. All this comes because we know now, we and others have been exploring LSD1 as an interesting target in oncology in the last 15 years. And we know now that LSD1 is really important in cancer cell stemness, and in some particular cancers playing a role in the transcriptional complexes governing the progression of this disease. So we have iadademstat. Iadademstat is the most potent and selective LSD1 inhibitor, which is in clinical development. We were recently publishing a paper comparing with other assets in development, and we know that the mechanism of action is basically dual. We know that it's controlling cancer stemness and differentiation in cancer cells, and we know that it's doing that by combination with different building blocks, transcriptional factors in different settings. I'm not going to extend on the details of the mechanism of action, but I will be very happy to provide you more insight if you are interested. So in leukemia, we and others characterize that LSD1 basically is involved in the differentiation blockade that produces the malignancy. And by inhibiting LSD1, we can hit leukemic stem cells and produce the differentiation of leukemic cells. So we have been in the last 4 years running ALICE. ALICE was started in AML first line in combination with azacitidine before the approval of venetoclax. And what we have seen, and we are going to present the last data of hopefully the final data, this ASH conference in New Orleans this December, is a consistent, very strong activity, more or less at the same levels that venetoclax, of course, different sizes of trials, with around 80% of objective responses, around 2/3 of them CR/CRIs, and very durable responses. the tolerability of the drug is quite good, and we have seen really the potential to combine this drug with many others. Okay, so that was very compelling, but the situation is that right now the market is controlled by venetoclax. So, I mean, to run against venetoclax or to try to do a triple combination would be a complicated task for a company like us. So our medical team in Boston has set up a new strategy, which is based on a second-line trial in patients harboring the FLT3 mutations, which represents 40% of the total population and represents a very interesting market, as you can see by the provisions of this is only a US market in terms of gilteritinib, which is occupying this space. So these patients are right now being treated with gilteritinib and FLT3 inhibitors, but still these patients are having a very strong unmet medical need. This is what we call a low-hanging fruit, an incremental improvement for those patients. And what we and others have been able to prove is that the combination of gilteritinib plus iadademstat produce a very strong synergism. So based on that, we proposed the FDA a clinical trial, which is FRIDA. FRIDA was approved by the FDA. Is going to be a phase 1b in which we are going to basically evaluate the tolerability of the combination and establish the recommended phase 2 dose. Okay, this is a program that plans to have as a secondary objective CR rates and the duration of response to enroll between 35 to 50 patients, and we are expecting to enroll the first patient any day now. So this is a program which is being led by the Massachusetts General Hospital, Dr. Amir Fathi, which is leading the program, the leukemic program there, and at the Dana-Farber is the principal investigator, but there are other centers like MD Anderson and some others which are also participating. It's a US-only trial. So the other indications where LSD1 plays a very important role are neuroendocrine tumors, and specifically small cell lung cancer is one tumor where there is still a strong unmet medical need despite the recent approvals of durva or atezolizumab, which have been providing only, again, incremental benefit to those patients. Why LSD1 is meaningful to combine here? For two reasons. First, we know, and there is a beautiful paper from the MD Anderson, that around 40% of small cell lung cancer are basically depending on the NOX1 signaling pathway, and LSD1 is able to restore the expression of the NOX1 signaling pathway, producing a response on those specific subset of small cell lung cancer tumors. But second, and very importantly, we and others, and there is a beautiful paper from Charles Rudin at the Memorial very recently using our compound, have shown that LSD1 inhibition, iadademstat, our compound, is able basically to boost the immunological response. And it's able to do this basically by a set of mechanisms. The first one is basically enhancing the expression of MHC1 genes, improving the interferon signaling pathway, producing that the tumor cells are being presented to the T cells. And not only that, we have seen that LSD1 inhibition, our compound, is able to produce a persistent duration, avoiding the exhaustion of the CD8 plus T cells, providing a more sustained response. So with all these strong fundaments, and taking into consideration that we had in the past previous clinical data on this indication, we are right now planning to do STELLAR. STELLAR is going to be a randomized control phase 1b/2 study in a small cell lung cancer in combination with a checkpoint inhibitor. Is a trial which is going to be led by the Foss Chase Cancer Center in Philadelphia, and we believe that this trial could potentially support an accelerated approval if we get a significant clinical benefit in those patients. There is another trial that we are about to start. This is a collaborative trial and a sponsor-initiated trial by the Fox Chase Cancer Center in second line in refractory relapsed neuroendocrine tumors. It's a basket trial, so it's an exploratory trial to identify which of these tumors could be more amenable to a combination in this refractory relapse setting. So this is has been submitted to the FDA, and we are expecting the IND clearance any day now. So this is the first pillar of the activities of the company. So as you have seen, we have a very ambitious program in oncology, but on top of that, we have a very novel, I would say that, again, we are the one and only company exploring epigenetics in CNS. And this is basically due to the fascinating fact that LSD1, as I said, is a master transcriptional gene, which is involved in early development, which is involved in the functioning also of the central nervous system and some other important systems. So we know now that there are evidences to play for a role, to advocate for a role for LSD1 in large multifactorial indications, but also in small rare monogenic indications, which is opening a very fascinating avenue. So in the characterization of vafidemstat, which is a second uncorrelated drug, LSD1 inhibitor, also very brain penetrant, safe in humans, and with a very nice pharmacology. In the early preclinical characterization, we characterize that LSD1 inhibition is basically able to reduce aggressivity, to increase sociability, and to enhance memory and executive functions. So with all this data that we and others have been consistently providing, we have done a number of exploratory clinical trials in the field. I'm not going to talk about that now, but we will focus today in basically on the psychiatric trial. So we did a basket trial in patients of autism, ADHD, or borderline personality disorder that were having a common factor, which was high aggressivity. And what we saw in this open label exploratory trial was basically that we were able to produce a significant reduction on the aggression, but also a significant reduction in the case of the BPD patients on the overall degree of the disease. So with all these data, we thought that the best route to market on these indications for us would be borderline personality disorder. Borderline personality disorders is a very prevalent and impairing disease, has very unstable and extreme symptoms, including aggression, self-aggression, and suicidal ideation and execution. So there is no right now any drugs approved on this field, and we know that Buffy is not only improving these symptoms in preclinical setting, but also in human patients. So competition is limited. So we think that we have a very good opportunity here with Portico. Portico is a phase 2b trial, basically being run in the US and in several countries of Europe. We plan to enroll about 150 patients, and we have two primary endpoints. One is the improvement in aggression. The second one is overall improvement in the disease. We are going to perform an interim analysis to calculate the signal size and futility around this first quarter, and assuming that we get the same current accrual expectations, we expect to have the final readout at the end of this year 2023. So that would be a transformational event for the company if we get the positive results here. We are running another earlier trial, which is being run so far only in Spain, evolution in schizophrenia, in cognitive impairment associated symptoms, and also in negative symptoms. I'm not going to explain more details now because this is an earlier trial, but I would like to spend a few minutes on our personalized medicine approach, which is a new concept in CNS. So this is basically following a bit the tracks of what we have been doing on the last years in oncology. So we know that there are a number of neurodevelopmental syndromes which are caused by monogenic disorders. Some of these monogenic failures are basically affecting genes involved in methylation and demethylation of chromatin, And importantly, some of these genes are the same that are happening in leukemia, and in which we are inhibiting LSD1, making a walls here. So we and others have shown that basically in some specific animal models, inhibiting LSD1 is able to mitigate or reduce or almost make disappear the symptomatology that we see in infants, in human children that are harboring these diseases. And Kabuki syndrome is a beautiful example. Kabuki syndrome is produced by a mutation in one histone methyltransferase, which is doing exactly the opposite job than LSD1. And among the symptoms that is produced in animals, what you see here is a lack of neurogenesis in the hippocampus. When these animals are being treated with an LSD1 inhibitor, these animals are able to produce neurons again in the hippocampus. So not only this capability to recover neurological expression, but also memory and others also some additional immunological insufficiencies. So that's why we have been talking and working, collaborating with the KOLs in the US at the Johns Hopkins, the Kennedy Krieger, or the Boston Children's Hospital to develop, to set up HOPE. HOPE is going to be an adaptive phase 1-2 trial in adults and in young adolescents, in which we want to see if the drug is well tolerated and if we are able to produce neurological improvements on those adults and young kids. So, of course, all this work that I have been mentioning here today in CNS and in oncology is done based on collaborations with the big institutions in the US and in Europe, and is basically being pushed by a management team with proven drug development and operational capabilities. As I mentioned, our medical team is based in Boston. We have our chief medical officer for CNS in San Diego, and Sai is our global chief business officer and investor relations based in New York. So with all this, we think that we have the right team and the right purpose. We have, again, a unique dual epigenetic proposition with aspects in CNS and oncology, with 2 programs that have been de-risked, 2 programs that have shown clear trends of clinical efficacy, and with value inflection points coming in the next 1-2 years, in which we are going to finalize ALICE. We are going to be able to show not only tolerability, but also efficacy in the combination with gilteritinib. We are going to be able to make the readout of Portico in borderline personality disorders, and starting new trials in Kabuki syndrome, in small cell lung cancer, and in neuroendocrine tumors, which are going to provide the second wave of catalysts for this company. With that, I will be very happy to take any questions. Thank you very much for your attendance today.
Thank you, Carlos. Thank you very much.